Carl June is an astonishing medical doctor and outstanding researcher at the University of Pennsylvania, known for his infamous pioneered work on cancer and HIV gene therapy. One of his most recent development, chimeric antigen receptor T-cell therapy, also known as CAR-T, was the first FDA-approved personalized cellular therapy for cancer in 2017. This therapy may seem to be a treatment from a science fiction novel because of the fascinating underlying science behind it. Cancers are complex diseases with individual defining characteristics within each diagnosed cancer patient. CAR-T therapy begins with a process known as leukapheresis, in which the patient's white blood cells are collected intravenously. Within the concoction of white blood cells reside T cells. The T lymphocytes are the cells of interest and are therefore isolated. After isolation, researchers in the lab place them in growing media for their proliferation as well as their activation. Once activated, the introduction of a gene encoding for a specific chimeric antigen receptor is inserted via an adeno-associated virus. This gene will encode a protein that will be transcribed in the rough endoplasmic reticulum, make its way through the Golgi apparatus, where it will be packaged and delivered to the cell membrane. The chimeric antigen receptor has three distinct regions that play an important role in its function. The extracellular domain has specific sequences that will recognize only the cells of target, in this case the cancerous cells. Therefore, the cancer cells must first be genetically sequenced in order to determine a particular sequence of target for that domain to recognize and bind to, while at the same time not binding to the surrounding healthy cells. The transmembrane domain keeps the receptor at the T cell's membrane, serving as an anchor. When bound to the target, conformational changes cause the cytoplasmic tail to initiate a signaling cascade and thus eliciting an immune response. Usually, cancer cells evolve mechanisms to evade their clearance by the immune system, but this therapy reinforces the immune system to prevent such evasions. Clinical studies on leukemia patients have demonstrated promising results. Therefore, further studies must first be performed before this therapy can be used on a wider range of cancer patients. Dr. Carl June is not only known for the development of this amazing new therapy, he is also known and has been recognized for much more.
June has published more than 350 manuscripts and has received numerous prizes and honors, including election of the Institute of Medicine in 2012, the Paul Ehrlich and Ludwig Darmstädter Prize, the William B. Coley Award, the Karl Landsteiner Memorial Award from the AARB, the Richard V. Smalley Memorial Award for the Society of Immunotherapy of Cancer, the Philadelphia Award, the Hamden Award for Medical Research Excellence, and the Novartis Immunology Award for Cancer Cell Therapy Development. He was elected to the American Academy of Arts and Sciences in 2014. In 2017, he was named a Fellow of the American Association for Cancer Research Academy and received the David A. Karnofsky Memorial Award from the American Society of Clinical Oncology. June is a graduate of the U.S. Naval Academy, as well as Baylor College of Medicine in Houston. He received graduate training in immunology and malaria at the World Health Organization in Geneva, Switzerland, and postdoctoral training in transplantation biology at the Fred Hutchinson Cancer Research Center